All right, so we're going to get started, everyone. Um, hi, everyone. Welcome to this week's all virtual Tuesday lunch seminar, where we invite internal speakers, mainly from our department, to talk about their work and research. Today, we have Dr. Michelle Fearon, postdoctoral research fellow in the EEB department, working with Professor Meg Duffy. Michelle received her undergraduate degree in biology with ecology and evolution specialization and a minor in wildlife, fisheries, and conservation from the University of California, Davis in 2013. She received her PhD in ecology and evolutionary biology here at the University of Michigan in 2020, working under Professor Elizabeth Tibbetts. Michelle's diverse research interests include the ecology of multi-host pathogens, the effects of environmental change on disease dynamics, ecological modeling, conservation biology, and behavioral ecology, among others. In the Duffy lab, she's using the freshwater zooplankton Daphnia to investigate how resource quality and parasite infection influences host physiology and the role of host community and environment on patterns of multi-parasite epidemics in lakes. So today we will hear from her on resource quality and host genotype differentially impact Daphnia parasite interactions, and I will hand it over to Michelle.
Great, thank you so much. Welcome everyone. I'm really excited to share with you the results of an experiment that I completed last year about how variation in resource quality and host genotype impact host parasite interactions. I'm just starting to write up this project right now, so I'm really welcome any feedback that you all may have on the story and analyses that I've done so far and ways that it might be able to be improved. So when we think about the typical process of parasite infection for an environmentally transmitted parasite within a single host species, it goes something like this. The host encounters and becomes exposed to a parasite in its environment, potentially while it's foraging. Depending on the genetic and environmental factors that control susceptibility and the parasite dose, the host may become infected by that parasite. Then, as the parasite begins to replicate within the host, it interacts with the host's physiology and immune system, resulting in a tug of war for the host's available resources and ultimately to determining the yield of parasite propagules and the survival of that host. Once the parasite's transmission stages are released, that cycle can begin again with the exposure of a new host in the environment. However, resource quality can impact these host-parasite interactions, sometimes in complex and opposing ways. Resource quality can alter the host exposure rate to a parasite. For some organisms, larger animals that forage more may be more likely to encounter environmentally transmitted parasite. Or hosts may change how they forage based on the quality of the resources. Hosts may increase foraging to take advantage of a high-quality resource. Or alternatively, a host may need to increase foraging in response to the poor quality resource due to an inability to get adequate nutrients from that source. Additionally, hosts may utilize different kinds of resources in different contexts, and that might change their exposure rate to a parasite if the parasite tends to only be associated with a particular kind of resource. Once the parasite is actually infecting the host, resource quality may influence the host's ability to upregulate energetically costly physiological or immunological defenses to adequately respond to that infection. Additionally, plants and algae produce secondary metabolites or other toxic compounds that, when are consumed by the host, may directly affect the parasite and can even have medicinal effects in certain circumstances. Finally, once the parasite is actively replicating in the host and producing new infectious propagules, the host and parasite may be locked in a battle to utilize the host's available resources to their own ends. The parasite may exploit the host resources to increase the yield of those parasite propagules and can result in the reallocation of resources to body size growth to make more space for the parasite, its replication, or shutting off reproduction within the host. Additionally, the host may be able to utilize those resources to invest in immune function and potentially recovery, or mediate the effects of the parasite on its fitness. Furthermore, both different genotypes may respond to differently to both the parasite and differing resource qualities. So here I'm showing two different genotypes indicated by the different colored DNA. The interspecific variation in foraging rates may contribute to different levels of parasite exposure in the environment, and different genotypes may prefer slightly different kinds of resources, which could impact the rate of exposure and the resource quality that they're experiencing. After parasite exposure, we know that in many host parasite systems, there are genetic factors that control the susceptibility, where some genotypes are highly susceptible, while others are generally much more resistant. Yet we often don't fully understand the underlying mechanisms that control whether a genotype is susceptible or resistant to infection. Different genotypes may shift how they allocate their available resources to reproduction, growth, and survival in different ways. In other words, they may shift their life history strategies in response to parasites to potentially resist infection, but likely with other costs to their host fitness. Additionally, different genotypes may vary in how sensitive they are to changing environmental factors, such as resource quality, which could limit the host's ability to reallocate resources when necessary. The complicated tug of war for the host's available resources will determine the outcomes of this within host interactions and contribute to differences in parasite yield among these infected genotypes, as well as host mortality. Ultimately, in many host parasite systems, we see evidence that changing resource quality and variation among host genotypes may interact with each other to alter the processes that control infection in different ways. But we rarely have good mechanistic or physiological understandings of what is going on within the host to clearly explain the ultimate outcomes of the host and parasite interactions under different resource quality scenarios. So in thinking about these complex interactions between the resource quality, hosts, and their parasites, this led us to ask, how will changes in resource quality impact host fitness, physiology, and infection dynamics? To examine this question, we're using the Daphnia model system, Daphnia dentifera model system. So Daphnia dentifera are zooplankton grazers that live in freshwater lakes. They are an ideal model system because they can reproduce both sexually and asexually. So we can generate clonal lines of these different genotypes that can readily reproduce in the lab. In this photo, you can see the asexual embryos that are in this Daphnia's brood chamber that I've pointed out with the red arrow. Additionally, Daphnia are commonly infected by parasites in their lake ecosystems and within the lab, and we can visually identify infected Daphnia. So in this photo, I'm showing you an uninfected Daphnia on the top. You can see that her body is rather clear compared to the infected Daphnia below, who is infected with a fungus. And you can see her body cavity is white and filled with spores. Furthermore, Daphnia are important in lake food webs for grazing on phytoplankton, including green algae and cyanobacteria. In turn, Daphnia are an important food source for predatory invertebrates and planktivorous fish. In particular, we know that resource quality is highly relevant to Daphnia in their lake ecosystems because population densities fluctuate throughout the season based on the availability and abundance of different algal and cyanobacterial food resources. But some food resources are better for Daphnia than others. Recent predictions suggest that increasing nutrient runoff, pollution, and climate change are expected to give toxin-producing Microcystis, a bloom-forming cyanobacteria, a competitive advantage over other green algae. These harmful blooms can result in eutrophic lakes, like the photo on the top. You can see this bright green lake. and can produce large die-offs when lethal amounts of microcystin and other toxins are produced by the cyanobacterial blooms. These changes not only represent a shift in the types of phytoplankton present in these lakes, but from green algae to cyanobacteria, but also a shift in the quality of resources for the zooplankton grazers like Daphnia, since cyanobacteria are generally considered to be a lower quality food resource. Daphnia produce fewer offspring and grow more slowly when feeding on cyanobacteria like Microcystis compared to green algae. Though Microcystis may be a low quality food resource, experimental evidence has shown that it may also have medicinal effects on parasites that infect Daphnia. Therefore, changes in these resource quality levels are likely to have downstream effects on Daphnia population densities and the infection dynamics within these lakes. So in other words, it seems that a shift from the high quality green algae to the poor quality cyanobacteria in lakes will be bad for the Daphnia hosts and bad for their parasites. Which begs the question then, how reducing resource quality in lakes will mechanistically impact interactions between Daphnia hosts and their parasites. Understanding the answer to this question will be important for the maintenance of lake ecosystems under current and future levels of human-caused environmental change to natural systems through nutrient runoff and climate change. In this study, we used a fungal parasite called Metschnikowia bicuspidata that infects Daphnia hosts in lakes to explore whether resource quality will impact these hosts' interactions with parasites, as well as among different genotypes in similar or different ways. So this fungus is, which I'll be referring to as the fungus for the rest of the talk, by the way. So this fungus is an obligate killer parasite. It is environmentally transmitted. So the Daphnia will be foraging by filter feeding phytoplankton in the lake, in the water column, and they will simultaneously ingest fungus spores while they're doing that. And these spores will then enter the gut of the Daphnia, puncture through the host gut wall, and then begin to proliferate within the host body cavity using the host resources. So here again, I'm showing you a picture where there's an uninfected Daphnia on the top and an infected Daphnia on the bottom, where you can see that her body is full of these fungal spores that are replicating within her body cavity. This fungus will cause host death within 10 to 16 days post-exposure, and then the spores will be released back into the water column upon the host death as the body cavity breaks open, and the spores will be released. So we set up an experiment that used a resource quality gradient from the high quality green algae Senodesmus to a low quality cyanobacteria Microcystis with a 50-50 mixture of the two to achieve an intermediate resource quality. The Microcystis strain that we used in this experiment does not produce microcystin toxin, but since microcystin toxin is commonly produced in harmful Microcystis blooms in natural lakes, we also included a toxin spike treatment with 15 micrograms per liter of microcystin, which will be that treatment will be shown in blue. So the 100% Microcystis and the Microcystis plus toxin treatments represent the same low resource quality level, but the comparison between these two treatments will allow us to specifically evaluate the effects of the toxin on host physiology and infection. However, it's a spoiler for the upcoming results. We generally did not see any significant differences between the M and the M plus toxin treatments due to just the greater impacts of the reduced resource quality in our experiment. So you'll see both of these results throughout the talk in the graphs I'm going to show, but I'm not really going to be talking much about these toxin results just because we really didn't see that many differences. But I'm happy to talk more about the Microcystis treatment and those results more afterwards if anyone's interested. And I will be using this color scheme and these letter labels. So Senodesmus will be S, SM for the 50-50 treatment, M for 100% Microcystis, and M plus for the Microcystis plus toxin treatment, along with this color scheme throughout the rest of the talk. We used a factorial design that included these 4 diet treatments and 2 host genotypes, mid-37 and standard, and the fungus, either a fungus exposed or an unaffected control treatment, for a total of 16 treatment combinations with 10 replicate animals placed individually in 50 ml beakers. So I think it's important to use 2 different host clones within this experiment because different genotypes vary in their susceptibility to parasites. They can invest in slightly different life history strategies to parasite, just in general, and may respond differently to these resource quality treatments as well. So ironically, our quote standard genotype is often anything but standard, as it tends to produce a large number of offspring and has a shorter lifespan, and is also tends to be highly susceptible to this fungal parasite compared to the mid-37 genotype we used here, as well as many of the other genotypes that we have in the lab. So to begin this experiment, we collected all of these animals as babies, then fed them the high quality Synodesmus diet for 2 days to make sure that they got to an adequate body size and there was no differences across these babies. Then we fed them their assigned diet treatment for 2 more days, and then we exposed them to the parasites or a sham exposure for 2 days. After that, we maintained all of the animals on their treatment diets until the end of the experiment at 24 days. We then measured a lot of things in this experiment. So we have measured the exposure rates of the parasite to these hosts under the different treatment resource quality treatments, parasite prevalence to get at susceptibility of these hosts to the parasite, and parasite yield. So this is the number of mature spores that are produced by each host. And then we measured a variety of physiological and fitness traits within the host. So that included the host hemocyte immune response to infection, the host feeding rates, host fecundity, which we also calculated using that data to calculate the population growth rate, little r. We calculated a body size growth rate, the host respiration rate, and host lifespan. So we expected that reducing resource quality could either positively or negatively impact the terminal infection prevalence. So these are the animals that produced mature fungus spores. The animals that are fed high quality diets may ingest more spores through higher feeding rates, or alternatively, high quality diets may allow animals to resist infection through upregulating their immune function. Before we dive too much into the data, I first want to take a moment to orient you to the figures that we're going to be looking at, because there will be many fairly similar looking graphs. So on the y-axis here, I'm showing the percent of animals that were infected with the fungus as the response variable. This will obviously change as I go through different metrics that we measured in our experiment. Along the x-axis, we have the 4 diet treatments, which will be shown as either points or box plots, and will be color-coded according to the legend, with light green for the high quality Synodesmus diet to dark green for the low quality Microcystis, and the 50-50 treatment is intermediate between those colors. And then the spike toxin treatment, M+ is in blue. The data will also be split based on each host clone, mid-37 and standard, so that you can see the similarities or differences between these 2 clones and how they respond to either parasites or the resource quality treatment. And finally, I'll include an ANOVA table of the results in the bottom right corner to indicate the significant treatment effects for those that are interested. So we found a strong effect of diet on fungus infection prevalence, where animals that were fed the high quality diets were more likely to be infected. And importantly, diets with Microcystis protected hosts from infection by the fungal parasite. So even the 50-50 diets were strongly protecting hosts from infection. The standard genotype tended to be a bit more susceptible to the fungus than the mid-37, but each genotype showed the same overall pattern with diet. So adding more Microcystis to the diets of these animals reducing the resource quality led to a negative effect on terminal infection prevalence for both genotypes. We hypothesized that this pattern may be explained by differences in the fungus parasite exposure. We measured the number of attacking fungus spores that either became embedded or penetrated the host gut wall as the parasite exposure rate here. We expected that the exposure data would mirror the terminal infection prevalence data that I just showed you, but it could be mediated by host behavior such as the relative feeding rate during parasite exposure. So animals in high quality diets may have higher feeding rates compared to animals with poor quality diets that may be trying to wait out that yucky food. However, after the spores attack the host's immune system may also impact the outcomes of infection. The host diet may impact the host's ability to upregulate costly immune responses to resist that infection. And so we measured the number of hemocytes, which is a type of immune cell that were attached to the attacking fungal spores. So first, looking at the feeding rate data, I measured the relative body size corrected feeding rate for each treatment during parasite exposure period for the uninfected and parasite exposed animals. And then I separated the parasite treatment animals by whether or not they were exposed but then resisted infection. So you'll see that as the exposed category on the x-axis, or they ultimately developed a terminal infection. And so those are the infected class and the parasite exposure x-axis here. So while the relative feeding rate did significantly decline with poor quality diets in the uninfected, unexposed animals on the left side of both of these panels, we saw that there was actually no difference in the feeding rate based on diet in animals that were exposed but resisted infection or those that ultimately became infected. Yet, despite there not being any difference in the feeding rates among these diet treatments for exposed animals to the fungal spores, I did find that diet still had a very strong effect on the exposure rates of attacking fungal spores that either embedded or punctured into the host gut wall. So animals that received these high quality diets in light green had many more spores attacking their guts compared to animals given the diets with any amount of Microcystis in it. Additionally, the standard clone tended to be more susceptible to the attacking fungal spores compared to the mid-37s. One of our collaborators tends to joke that the standard guts are just like butter for these parasites, so the spores can just go right through them. The results mirror the infection prevalence results that I've already shown you, but they are not caused by differences in the feeding rate nor differences in body size among these animals. So there must be something else that's going on that's changing the number of attacking spores. We can visualize these patterns very clearly under the microscope at 40x, where animals on the left that were fed the high quality Synodesmus diet have many more fungus spores attacking their gut wall, indicated by all these red arrows. Each of those red arrows is pointing to a needle-like fungus spore that has either embedded or punctured through the gut wall. Meanwhile, on the right, the animal that is fed the low-quality Microcystis diet had very few or no attacking spores in their guts one day after parasite exposure. So here you can see these are the muscles of the Daphnia, but there are no needle-like spores going through this gut wall here compared to many, many spores puncturing through this gut wall on the Synodesmus fed animal. So Daphnia with attacking fungus spores can recruit hemocytes, a type of immune cell, to the site of infection to try to kill those parasites. So within this blue box, you can see that some of the needle-like fungus spores have already punctured through the gut wall and have a number of hemocyte cells attached to them. And so we wondered whether diet might influence the host immune response to infection. We found that significantly more hemocytes were recruited to attacking spores in animals that were fed the high quality diet compared to all other diets. And there were no differences among the two host clones. However, since I previously showed you that the high quality diets also had a lot more attacking spores, we calculated the number of recruited hemocytes per attacking spore to better see if diet quality might be influencing the host's immune response to parasite exposure and infection, since immune responses tend to be energetically costly. Once we took the number of attacking spores into account, there was no significant differences among the treatments nor the two host clones. So this suggests that the host's immune capacity is simply responding to the intensity of parasite exposure rather than altering their immune responses based on the quality of their resources. And the immune response does not appear to be altering the outcomes of terminal infection. So while diet quality did impact both fungus exposure and infection, feeding rates were not different based on the diet quality treatments in exposed animals, and immune responses were consistent regardless of diet quality in these parasite exposed animals. So what is causing these strong diet effects on parasite exposure and infection? Another alternative explanation for the diet effects that we've observed here could be differences in the host digestive enzymes that are activated or inactivated based on the different diets that they're foraging on, and maybe also influencing the likelihood of the spores attacking the host. So some exciting work from my lab mate, Crystal Sanchez, has shown that dietary effects on digestive enzymes may be the key to these differences in fungus parasite exposure and infection. Her hypothesis was that as the Daphnia are feeding, they will consume the spores along with their food. And these different food types may induce changes in the expression of gut enzymes within their gut and that are used to then digest their food, and may also be interacting with the spores in their gut to allow for dehiscenc, which is a process where the spore loses its protective ascus and allows the needle-like spore to then puncture the host gut wall and then continue to infect the host. However, if the gut enzymes are blocked by some aspect of the diet, then the spore is not able to lose its ascus and cannot then infect the host. So in an experiment, Crystal found that many Daphnia hosts that are fed the same Microcystis strain that I used in my study, which is shown here in blue, they did not produce any of this chymotrypsin digestive enzyme. You can see all of the Microcystis treatments have zero chymotrypsin activity, compared to a Sunidesmus diet, also the same one that I used in my experiment, which has normal levels of chymotrypsin activity along the x-axis here. Additionally, she also found that fungus infection was either the same or higher in the Sunidesmus fed animals compared to the Microcystis animals. And you can see that by these lines are either going straight across or going up. So to further test the impact of diet and digestive enzymes on the dehiscence process, she conducted an experiment that measured diet effects on removing the spore's ascus within the Daphnia guts, which I'm going to show on this figure on the left. So if the spore does not lose its ascus, then it cannot infect the host. So on this y-axis here, if there are treatments that fall in this lower category here, that means that those spores are not losing their ascus and would not be able to infect the hosts, whereas treatments that fall on this higher category up here on the y-axis have lost their ascus and are much more likely to be able to infect the host. So in her experiment, the treatments included a Sunidesmus alone, a Microcystis alone, a chymotrypsin extract alone, and then a Microcystis combined with that chymotrypsin extract to see if adding the chymotrypsin back in would increase the likelihood of removing the spore's ascus and a buffer control. So the Synodesmus-fed animals had higher numbers of spores without an ascus, while the Microcystis diet had very few spores that had the ascus removed. So again, that means the Microcystis-fed animals are much less likely to be able to infect the hosts. The chymotrypsin extract only diet dramatically increased the number of spores without an ascus, and the combination of Microcystis with the chymotrypsin ascus recovered the ability to remove the ascus spore, the spore's ascus compared to the Microcystis treatment alone. So altogether, Crystal's data is consistent with the parasite exposure patterns that I observed in my experiment and suggests that Microcystis diets inhibit chymotrypsin enzymes in the host gut. That is crucial for not only digesting their food, but also the dehiscence of the fungus spores in the gut to allow for penetration and infection. So next we looked at whether reducing the resource quality might impact the parasite yield within these animals that did get infected. So these are the animals that produced the number of mature spores that these animals produced. We expected that reducing resource quality would reduce the parasite's capacity to proliferate because there would be less host resources for the parasite to then exploit. Overall, we found relatively little variation among the infected animals for spore yield, but we did see a moderate effect of diet on spore yield here. So it was just significant with a p-value of 0.048. So we observed a marginal effect of host diet on the number of mature spores that were produced. So looking at these animals themselves is actually shows a consistent pattern with the infection results that I've already shown you. So these two animals are the same age and the same genotype, and both were exposed to the fungal parasite. But they were either fed a Synodesmus diet on the top or a Microcystis diet on the bottom. The Synodesmus-fed animal is opaque and full of fungal spores, while the Microcystis-fed animal has been protected from infection. You can see that her body is quite clear. However, diet can also have strong impacts on host fitness and physiology, and we need to consider these factors in order to understand the full effects of resource quality on hosts when they're in the presence or absence of parasites. For example, you can see here that the Synodesmus-fed animal has grown much larger than the Microcystis-fed animal, but the Microcystis-fed animal is still reproducing. You can see her one embryo in her brood chamber here. While the Synodesmus-fed animal, her brood chamber is empty, but it's quite large, which indicates that she previously did produce potentially a large number of offspring early on before the onset of her symptoms, but now she is no longer able to reproduce due to her infection. So going back to this concept diagram, hosts may alter the allocation of their available resources into reproduction, growth, metabolism, or survival. Or in other words, they could shift their life history strategy when they're experiencing different levels of resource quality in the environment. In turn, these life history shifts may influence whether the host becomes infected or is able to resist infection when they're exposed to parasites. In general, we found that reducing resource quality negatively impacted host reproduction, growth, and metabolism, but not overall survival in uninfected, unexposed animals. So essentially, we have proven that our poor quality diet is in fact poor quality for these hosts. We know that these poor quality resources are bad for the host and bad for the parasite, but what happens when these two factors co-occur? How do the hosts deal with these challenges? So I'm going to show you just a subset of these results for the life history traits, just for the sake of time, because this experiment had so much data. But I'm going to primarily focus on the differences between the uninfected hosts and the hosts that were exposed to the parasite but resisted infection to demonstrate some of the more interesting life history shifts that appear to signal costs of reducing resource quality and parasite exposure. So in general, we expected that if there was a cost to resisting parasite infection, we would see a reduction in the key life history traits. So I'm showing that in this figure here, a reduction in the life history trait in the exposed but remaining uninfected animals compared to the unexposed and uninfected animals. However, different host genotypes may alter their life history traits in different ways, and they may alter those same life history traits to differing degrees based on their diet quality or just based on genotype. So we used our fecundity data to calculate the instantaneous per capita population growth rate per day, also known as little r, for each diet quality treatment, host clone, and infection status combination. So this metric estimates the population's growth for a given host genotype and under the diet conditions that for that treatment. This calculation produces a single little r estimate with bootstrap standard error. So we can use those confidence intervals to evaluate differences between these treatment combinations. We found that standards generally had a higher little r compared to the mid-37s. And there was a steep decline in little r based on the diet quality, which you can see for each infection class here, that generally declines based on diet quality. Since little r heavily weights early reproduction, we don't actually see quite as large differences between the uninfected and the infected animals as you might expect, because the infected animals were still able to produce their first clutch prior to the onset of symptoms. So they had at least a little bit of reproduction before they became too symptomatic and stopped reproducing. However, when we compare the uninfected animals to those that were exposed but resisted infection, we can see some interesting patterns. So I've drawn a little cartoon up in this upper right corner to show the key takeaways for this uninfected compared to exposed but resisted infection animals. And you can get those main takeaways from that. But so standards exposed to the fungus had a lower little r than the uninfected standards across the 3 lower quality diet treatments shown in this red box here. And similarly, the mid-37s that were fed a high quality diet and exposed to the fungus seemed to have a lot lower little r compared to the corresponding uninfected treatment, but there was no change in little r among the mid-37s that were fed any of the 3 poorer quality diets. So I expect that they were already so affected by their poor quality diet, reducing their reproduction, that the parasite exposure didn't seem to change that for those treatments. We calculated the host body size growth per day from during parasite exposure to the following week after exposure, which varied for each diet quality, infection status, and host clone treatment. In general, host growth rate declined with the poorer quality diets and was lower in the infected animals compared to uninfected animals. And it's a little hard to see, but on average, standards grew a little bit faster than the mid-37s did. So when I analyzed the data, excluding the infected animals to only compare the unexposed, uninfected host to the parasite exposed but resisting infection, I found a marginally significant infection by clone interaction. So parasite exposed mid-37s grew marginally less compared to the unexposed, uninfected hosts, suggesting a slight cost of parasite exposure in the mid-37s, but there was no difference in growth based on the parasite exposure for these standards. Finally, we evaluated how the treatments impacted host lifespan and risk of death. On the right, I'm showing a forest plot of the Cox proportional hazards model that calculates the risk of death, where hazard ratios that are greater than 1 increase the risk of death, and hazard ratios that are less than 1 indicates a decreased risk of death. So on the left, I have a box plot of the lifespan of the animals based on their diet, infection status, and host clone treatments. Animals that lived to the end of the experiment were censored at 24 days, and you can see that most of the uninfected animals for both genotypes lived to that maximum 24 days. So that's why it kind of looks capped off here at the top. Unsurprisingly, the infected animals had a greater risk of death, but interestingly, animals that were exposed to the fungal parasite but resisted infection died even faster than the infected animals. So these results suggest that fungus exposed animals are definitely suffering a cost to resisting infection. We also found an interesting interaction between diet and infection status, where infected and exposed animals that were fed the high quality diets tend to die even faster than the animals in the lower quality diets. Additionally, we saw that the standards tended to die earlier than the mid-37s in this experiment, further suggesting that the standards in the mid-37s have slightly different life history strategies. So the standards tend to grow faster, reproduce more offspring early, but they don't live as long compared to the mid-37s that produced fewer offspring, grew more slowly, and lived longer. To review the main results from the experiment, we'll first look at how diet and host genotype influenced parasite exposure, infection, and yield of new infectious propagules, where I'm going to show a check mark if there was a significant difference, or an X if there was not, and an arrow indicating the main direction of the effect of either increasing or decreasing with declining resource quality. So diet quality and host genotype both strongly influenced host exposure to attacking spores, where animals that were fed the high quality diets became highly exposed to the fungal parasite, while animals fed the lower quality diets were protected from spores attacking their guts. Our data corroborates and builds upon previous work from the lab that showed that low quality resources decreased fungal transmission potential by stunting host growth and reducing feeding rate. Our results further show that the lower infection prevalence in poor quality resources is due to the fungal spores not attacking the gut at the same rate as the high quality resource treatments, despite receiving the same spore dose and having similar feeding rates on these different diets. So, and then furthermore, we also show that the host immune response is a symptom of infection rather than an effective response, and did not change based on the resource quality that they were experiencing. Based on the large differences in parasite exposure, we consequently observed that high quality diets had very high fungal infection prevalence compared to low quality diets. And diet quality had a weak but significant effect on spore yield, but there was no difference between the two host clones in spore yields. Previous work in the lab has found similar infection prevalence based on these diet qualities, including a potential medicinal effect of the microcystin toxin, but found no difference in the fungus spore yield between the high quality Cynodesmus and the low quality Microcystis diets within a single host clone. In my experiment, the Microcystis diet strongly protected hosts from infection, so infection prevalence was too low in the M and the M+ treatments to be able to assess whether the toxin treatment had a medicinal effect here. So it's possible that it still did, but we just weren't able to assess that in this experiment. Next, we'll look at these factors from the host perspective to see how resource quality and infection size affect the host feeding rate, population growth rate, body size growth rate, and survival. So we found that host feeding rate only declined with poor quality diets for uninfected hosts, while parasite exposed or infected hosts had slightly higher feeding rates, but were not affected by their diet. The host population growth rate and the body size growth rates both declined with poor quality diets and in infected animals compared to uninfected animals. Resource quality did not impact host survival in uninfected animals, but had an interesting interaction with infection status, where infected animals fed the poorer quality diets lived longer than the animals in higher quality diets. Hosts that were infected by the parasite died earlier than uninfected hosts, and parasite-exposed hosts that resisted infection died faster than both of those groups. Importantly, many of the host life history traits that we measured also differed between the two host genotypes in our study. On average, standards had a live fast, die young life strategy where they tended to produce more offspring, grow faster, and have a reduced survival compared to the mid-37 clones. But one of the really interesting results of this experiment was how the different host genotypes responded to exposure to the fungus parasite. So compared to the unexposed, uninfected hosts, each genotype shifted their feeding behavior and several aspects of their life history when they were exposed but resisted infection. The exposed mid-37s had higher feeding rates than uninfected mid-37s, but standards did not change their feeding behavior based on the parasite presence. Only mid-37s fed the highest quality diet, reduced their population growth rate, indicating lower offspring production when exposed to the parasite, but hosts fed the poor quality diets already had such low population growth rates that there was little to change between the uninfected and the exposed hosts. On the other hand, for standards, we observed significant declines in population growth rates in the parasite exposed hosts compared to the uninfected hosts for all of the diets. Mid-37s also marginally reduced their body size growth rate when exposed to parasites, but we did not see that same effect for standards. And finally, across all diets, we found that standards had a larger reduction in survival than mid-37s, but both host genotypes had reduced survival when they resisted infection compared to hosts that were not exposed to parasites. So altogether, this data shows that there are significant costs for hosts that resist infection, but each genotype responds to this challenge differently, apparently by allocating their resources in different ways and shifting their life history strategies. So I also did the same experiment with a second bacteria parasite that is ecologically quite similar to the fungal parasite that I used here. And although I did not have time to show you all of that data in detail, I do want to briefly highlight the differences that we saw between the genotypes in each parasite species. So here, don't worry too much about what these different arrows might mean. The main thing I want you to notice is that the arrows differ based on both the host genotype and each parasite that they were exposed to. So this highlights the potential variation that exists in how individuals differentially respond to multiple parasites in the environment and the complex ways that hosts may allocate their potentially limited resources to alter susceptibility to those parasites. To briefly summarize the findings from the study, we show that resource quality and host genotype strongly impacted host exposure and infection rates to the parasite, where animals in the high quality resource treatments experienced much higher exposure and infection prevalence compared to the poor quality resource environments. But neither feeding rate nor immune function mediated those differences in exposure or infection. When comparing the uninfected and unexposed hosts to those that were exposed but resisted infection, we found that diet quality consistently reduced host fecundity, growth, and survival, and each host genotype changed their feeding behavior and life history strategies in different ways when exposed to parasites. It appears that each genotype differs in how it allocated their resources and the severity of the fitness costs to resisting parasite infection, which ultimately contributed to different outcomes in infection and host mortality. So overall, life would be easier if these patterns were consistent across genotypes, but that is not the reality. And we need to acknowledge that there's intraspecific variation in each step of this infection process and how different genotypes respond to the resource quality in their environment. In this study, we only examined 2 genotypes, and yet we still see that each responded in unique ways to these diet effects and parasite exposure. Therefore, it's essential to conduct studies with multiple genotypes in order to better understand the diversity of host responses that may occur to changing environments and their potential impacts on population dynamics in natural systems. So I owe a huge thanks for this work to the entire Duffy lab, because this was one of those experiments that really requires the entire lab's help to run. So I particularly want to shout out Crystal Sanchez, Sun, Kira, Siobhan, and Varun, who is an undergraduate that worked on this project, they significantly contributed to this experiment and its design. I'd like to thank John Megahan for the cartoons that I used throughout this talk, as well as the Moore Foundation and NSF for their funding. With that, I'd be happy to take any questions based on this work and thoughts on ways that it could be improved. Thank you. See, I can't see anything in the chat, so someone has questions. Maybe they can just unmute themselves.
I can ask one while other folks think of them. So in your dissertation research, you also were looking at food quality and effects on disease. And so I guess I'm curious about, like, if you merge this work and your dissertation work, do you have an overall kind of synthesis of the effects of resource quality on host parasite interactions? That's a big thing to ask you to do on the spot, but
yeah, that's a great question. And unfortunately, as is often in ecology, it seems that it's context dependent. So yeah, in my dissertation work, I found that certain metrics of resource quality impacted viral prevalence in bees in different ways and in different hosts. And so there it was often the landscape level amount of natural habitat, which could indicate a lot of different things. So that could be just overall abundance of natural resources, as well as diversity within a natural ecosystem relative to an agricultural ecosystem. And that abundance of the plants, flowering plants did also matter for some parasite host interactions. So I think it's kind of hard to come up with an overall impact of resource quality, though, because even here among the two, I didn't get to talk about the bacterial experiment as much, but we see really different patterns of resource quality for that parasite. So for the bacterial parasite that I similarly did here, diet effects did not matter as much for parasite infection and parasite yield. So we see quite different impacts depending on the host and depending on the parasite. So it's hard to come up with a generality. Again, I can't see the chat, so if there's any questions that are there, maybe someone could read them out to me. I can't seem to.
There's nothing there. Seems like maybe there aren't other questions.
All right.
Yeah, I suppose there aren't any more questions. So thank you so much for sharing your work, Michelle.
Thank you. I appreciate the opportunity to share it all with the department. It was a lot of fun to put this talk together.
Okay, thank you. And thank you everyone for tuning in.
Thank you. Bye.
Thanks, everyone.